They say, hold on to the ones you love. Keep them close to you. And they say, hold on to this time we have. And let the light shine through.
海の周りにおる方は宝物がいっぱいあるよって言ったように、言っておる方がおいでました、昔の方は。だから宝であるから大事にその宝を利用しないと価値があるよというようなことのであると思うんですけどもね。名前は、戸谷良一です。で職業は揚山製塩という形で製塩ですね、塩作り。こういった塩作りの製法というのは世界どこでもなく、この能登半島にしかないと。もうそうですね、伝統一応継承してますので400年以上のね、今年で23年になります。3トン半かける今やったこの年数かければ大体それはこれだけブーブで作ったのかなというような思いであるんですけども、大体朝ですね、4時半頃からここに来られて、そしてまず海水を汲み上げて、そして海水をまいて、大体僕らが聞いてるのは100坪の広さで約70リッターの桶を担いで、それが19回1トン200から大体120キロの塩までということです。砂が乾いたら寒さを集めて、そしてそれを箱の中に入れてから、また上から海水を注いで砂に付着している塩分を濃縮するわけですね。ミネラル成分が違うんじゃなかろうかというふうに私は思うんですけども、やっぱり鉄分かなんかそういうのが含まれているということで、ミネラルのありが違うんじゃなかろうかというふうに思うんですけども。大体6時間ほど煮詰めますと、1週間働かないとその600リットルのその海水、濃い塩水が取れないということですね。釜屋の中にむしろを引いて、そこに休みながらやってたんですよ。揚山の塩というのは海水を利用しますので、料理に使う塩ですね。味っていうのはまろやかなしょっぱみがあって、舌触りが滑らかで、一番僕はいいんですけども、おにぎりに一番適しているんじゃなかろうかというふうに思っています。皆さんいい塩を求める傾向があるということで、年間このほかに2カ所ありますので、僕の目指しているのは大体2カ所12トンほどは取れると思ってるんです。このちっちゃい頃からね、花が来ます、園児からどうして塩ができるのとか、この塩水が宝物やろうって、そういうふうに言ってるんだけど、
ලංකාවේ ජනගහනය වැඩි වෙනවත් එක්කම රෝඩ් ට්‍රැෆික් කියන එක වැඩි වෙලා තියෙනවා. ඉතින් එතකොට ඇක්සිඩන්ට් වෙන්න පුළුවන් ප්‍රමාණය වැඩි. හැබැයි මේ හැම එකකදීම ඉමීඩියට් ඇක්ෂන් එකක් ගන්න නැති අවස්ථාවන් එන්න පුළුවන්. ඉතින් ඒකට අපි හදලා තියෙනවා ඇක්සිඩන්ට් ඇලර්ට් ඇන්ඩ් ප්‍රිවෙන්ෂන් සිස්ටම් කියලා එකක්. මේකට අපි පාවිච්චි කරලා තියෙනවා වයිබ්‍රේෂන් සෙන්සර් එකක්, ඇල්කොහොල් සෙන්සර් එකක්, ෆ්ලේම් සෙන්සර් එකක්, ඕන බෝඩ් එකක්, GPS මොඩම් එකක්, මෙන්න මේ වගේ දේවල් පාවිච්චි කරලා තියෙනවා. මේකේ අපේ මේන්ම පර්පස් එක තමයි මොකක් හරි ඇක්සිඩන්ට් එකක් වුණ ගමන් ඒ උන අදාළ ඩ්‍රයිවර්ගේ පවුලේ කට්ටියට එහෙම නැත්නම් එයාගේ යාළුවෝ කාට හරි මෙන්න මේ ලොකේෂන් එකේදී ඇක්සිඩන්ට් එකක් වුණයි කියන එක මෙසේජ් එකක් මගින් දැනුවත් කරන එක. අපි මෙතනදි ඇල්කොහොල් සෙන්සර් එකක් පාවිච්චි කරලා තියෙනවනේ. ඉතින් මේකෙදි මුලින්ම වාහනේ ස්ටාර්ට් කරන්න කලින් ඩ්‍රයිවර් ඇල්කොහොල් පාවිච්චි කරලා තියෙනවද නැද්ද කියන එක මේකට පුළුවන් ඩිටෙක්ට් කරගන්න. ඩ්‍රයිවර් බ්‍රීත් කරන එක කැල්කියුලේට් කරලා ඇල්කොහොල් කොච්චර ප්‍රමාණයක් අරන් තියෙනවද කියන එක අරගෙන මෙයා ස්ක්‍රීන් එකේ ඩිස්ප්ලේ කරලා තියෙනවා driver is drunk එහෙම නැත්නම් driver is not drunk. ඊට පස්සේ you cannot drive එහෙම නැත්නම් you can't drive කියන එක. ඉතින් මේක සාමාන්‍ය ගොඩක් වෙලාවදී අපිට ප්‍රයෝජනවත් වෙන්නේ දැන් ටැක්සි එකකදී වගේ අපි දන්නේ නැහැ ටැක්සි ඩ්‍රයිවර් ඇල්කොහොල් අරන් තියෙනවද නැද්ද කියන එක. ඉතින් ඒක බලපානවනේ අපේ සේෆ්ටි එකට. ඒ නිසා මේක ප්‍රයෝජනවත් වෙනවා. ඊට පස්සේ මෙතනදී තියෙනවා ভাইබ්‍රේෂන් සෙන්සර් එකක්. දැන් අපේ වාහනේ ගිහිලා කොහෙරි වැදුනට පස්සේ ඒ ভাইබ්‍රේෂන් සෙන්සර් එක ඩිටෙක්ට් කරගන්නවා මේක ভাইබ්‍රේෂන් එකක් කියලා. ඊට පස්සේ ඒ ভাইබ්‍රේෂන් එක කැල්කියුලේට් කරලා ඒක ඇක්සිඩන්ට් එකක් නම් එයා තප්පර 30ක් බීප් වෙනවා. මේ සෙන්සර් එක තප්පර 30ක් සෙන්සර් එක මගින් තප්පර 30ක් බීප් වෙලා තප්පර 30කට පස්සේ අර අදාළ කන්ටැක්ට් නම්බර්ස් ඒ කියන්නේ ඩ්‍රයිවර්ට පුළුවන් තමන් ඇක්සිඩන්ට් එකක් වුණාම තමන් දැනුවත් කරන්න ඕන කාටද? තමන්ගේ යාළුවෙක්ට වෙන්න පුළුවන්. එහෙම නැත්නම් කාගේ හරි පවුල කෙනෙක්ට වෙන්න පුළුවන්. අන්න ඒ ඕන තරම් කන්ටැක්ට් නම්බර්ස් වලට මෙසේජ් එකක් යනවා GPS එකෙන් ලොකේෂන් එක ට්‍රැක් වෙලා මෙන්න මේ ලොකේෂන් එකේදී ඇක්සිඩන්ට් එකක් වුණයි කියන එක. හරියටම ඇක්සිඩන්ට් එකක් වෙලා තප්පර 30කට පස්සේ. ඒ වගේම තමයි දැන් මේ ඇක්සිඩන්ට් එකේදී ඩ්‍රයිවර්ට ඉන්ජරි එකක් නැත්නම් එයාට එහෙම ඕන නැත්නම් එහෙම කාටවත් මෙසේජ් එකක් දෙන්න ඩ්‍රයිවර්ට පුළුවන් මැනුවලි මේ මෙසේජස් යන එක නතර කරන්න. ඒක තමයි ভাইබ්‍රේෂන් සෙන්සර් එකෙන් කරන්නේ. ඊගාවට මෙතන තියෙනවා ෆ්ලේම් සෙන්සර් එකක්. මේ ෆ්ලේම් සෙන්සර් එකෙන් මේ ෆ්ලේම් සෙන්සර් එකෙන් වාහනේ ඇතුලේ මොකක් හරි ගින්නක් ඇති වුණොත් අර විදිහටම තප්පර 30ක් බීප් වෙලා මෙසේජ් එකක් එනවා ඩ්‍රයිවර්ට ෆයර් ඇක්සිඩන්ට් ඩිටෙක්ටඩ් කියලා. ඒව තමයි එතනදි වෙන්නේ. ඊට පස්සේ අර මම කිව්ව මෙසේජ් එක තමයි මේ තියෙන්නේ. මෙන්න මේ වගේ මෙසේජ් එකක් තමයි ඩ්‍රයිවර්ට ඒ වගේම පවුලේ කට්ටියට යන්නේ. අපි දැන් බලමු කොහොමද මේක වැඩ කරන්න කියලා මම පෙන්නන්නම්. මෙන්න මේ වගේ ভাইබ්‍රේෂන් එකක් ආවට පස්සේ මෙහෙම තප්පර 30ක් බීප් වෙනවා. ඊට පස්සේ ඇක්සිඩන්ට් ඩිටෙක්ටඩ් කියන එක කියනවා. මේකෙන් තප්පර 30ක් උනාට පස්සේ තමයි මෙසේජ් එක යන්නේ. මම ඒක මැනුවලි නවත්තලා පෙන්නන්නම්. ඊට පස්සේ ෆ්ලේම් සෙන්සර් එක වැඩ කරන්නේ මොකක් හරි ෆයර් ඩිටෙක්ෂන් එකකදී. අර ගින්දරක් ආපු ගමන් එයා ඩිටෙක්ට් කරගන්නවා ෆයර් ඇක්සිඩන්ට් එකක් කියලා. ඒක තමයි එතනදි වෙන්නේ. මේ ටික තමයි අපි දැනට හදලා තියෙන්නේ. ඊගවට අපි ඉදිරියේදී මේක ඩෙවලොප් කරන්න බලාපොරොත්තු වෙනවා. ඇක්සිඩන්ට් එකක් වුණ ගමන් ළඟම තියෙන හොස්පිටල් එක ඒ වගේම පොලිස් ස්ටේෂන් එක ඒගොල්ලන්ගේ ලොකේෂන් ට්‍රැක් කරලා අපිට පුළුවන් දැනුම් දෙන්න මෙහෙම ඇක්සිඩන්ට් එකක් වුණයි කියලා. එතකොට ඒ අදාළ ඉමීඩියට් ඇක්ෂන්ස් ගන්න පුළුවන් නිසා. ඒ වගේම තමයි අපිට පුළුවන් වාහනේට කැමරා මොඩියුල් එකක් හයි කරලා ඩ්‍රයිවර්ගේ බිහේවියර්ස් බලන්න. බිහේවියර්ස් චෙක් කරලා එයා ඇහිපිය ගහන රේට් එක කැල්කියුලේට් කරලා අපිට පුළුවන් ඩ්‍රයිවර් නිදිමතේද ඉන්නේ කොහොමද කියන එක. ඊට පස්සේ එයා ඩ්‍රයිවර්ට දැනුම් දෙන්න පුළුවන් ඩ්‍රයිවර් ඉන්නේ නිදිමතේ. ඒ කියන්නේ ඒක දැනුම් දීලා ඒකට අදාළ ඇක්ෂන් එකක් ගන්න පුළුවන් විදිහට අපිට මේක ප්‍රෝග්‍රෑම් කරලා ඩෙවලොප් කරන්න පුළුවන්. ඒ ටික තමයි අපි ඉදිරියේදී බලාපොරොත්තු වෙන්නේ. Thank you.
Billions of species have been wiped out in the face of perpetual aggressions of nature. Yet humans have always prevailed. They always knew how to adapt, innovate, and be defiant to circumvent the deadly challenges on their way. With the rising energy prices and inflation, declining food supply, soaring world population, unwarranted political conflicts, culture wars, and major environmental challenges, we are yet again at the crossroads. The present demands actions to reverse course and find sustainable solutions to save humanity and nature from the brink of total collapse. With 8 conference tracks and over 200 speakers, the ICSUSL 2023 sets the stage for scholars and scientific minds around the world to work together and create global partnerships to achieve the sustainable development goals set by the United Nations. Join us at the 9th International Conference of the Subrahmanya University of Sri Lanka as we celebrate research and progress of humanity to advance multidisciplinary approaches for a sustainable and resilient future.
Flensburg, Sweden. He received his PhD in molecular biology from Swedish University of Agricultural Sciences, or SLU, and has been a research scientist and a postdoctoral scientist at SLU, Lund University, Sweden, University of Copenhagen, Denmark, and Uppsala University, Sweden. He has been an associate professor in chemistry with specialization in molecular biophysics, Lund University, and also an honorary academic member in University of Colombo Institute of Biochemistry, Molecular Biology and Biotechnology. Since 2017, he has also become a member of R&D in leading innovator companies focused on cancer drug discovery and currently serves the Sprint Bioscience, a leader in producing innovative and breakthrough oncological drug candidates. Professor Ubesekara, again, let me be honored to invite you on stage for your talk.
Thank you for the introduction. And first, I would like to thank my classmate, my friend, Professor Nimal Udayakumara, for inviting me to give this lecture to you today. So let's begin the talk. So I am going to talk, I am going to discuss, if you are engaging with the discussion, about the structural biological applications and how to determine structures of large molecules, and so why are we interested in doing this, right? So it's the title, Molecular Machines, Protein Structure and Function. So I am going to briefly discuss these. Oh, I'm sorry, I was just shifting slides. Okay, so I will just briefly discuss why we need protein structures and the methods we use to solve the structures of proteins. And the main one of the methods, X-ray crystallography, I will discuss a little bit extensively. So I will give some examples. Then I will talk a little bit about homology modeling, which is another method of structural studies, and give some examples. Then I will just mention about drug design and enzyme engineering. So we understand biology in different levels. For example, if a zoologist understands biology completely different than a botanist, right? And there are cell biologists and people who work on parasites, people who work on fungi, bacteria, and infectious diseases, and cell biologists, and people like us understand biology in completely different way. We, for example, people like us, so like structural biologists or protein crystallographers, we try to understand how the molecules and atoms work. So we go down to the atomic level, or say it's fair to say we go down to molecular level, right? So just to briefly tell you what are macromolecules. So there are say 4 different types of macromolecules in living systems, like polymers of amino acids, they are proteins, polymers of nucleotides, DNA and RNA, then polymers of sugar, we know, like cellulose, like this wood, or starch, for example, and then the membranes, which are structural aggregates of lipids, like cell membrane and different other membranes. Why these macromolecules play a vital role in our life. So that's why we are studying these macromolecules. So why we need to determine or to know the structures of macromolecules? Because seeing is believing, right? If you see something, you know exactly what it is. So that's why we, to understand something, we have to determine the structure and then see it, how it looks like and how it functions, and you can obtain structural insights from that. I'll wait a couple of minutes until they sit. Okay, so let's continue. So to understand the activity of proteins, we need the structure. To design more useful enzymes or useful proteins, we need the structure. And for the drug designing, we need structures. So it may be like alien to you just to hear about starting from the structure of protein, right? But you will understand when I explain more. And if you have questions, you are welcome to ask me. So these are basic protein structural properties. These proteins, they are small machines, right? So for example, if you in a garment factory, there are machines for like cutting clothes and then sewing different parts of clothes. Similarly, when we eat, when we breathe, whatever we do, everything is taken care of by these small machines. They are called proteins. like enzymes, for example. They are small. Actually, enzyme is a small machine. So, and they binds to DNA, RNA, and other molecules. So they have space for binding space for other molecules. They can be rigid. No, they are not rigid, but they are flexible. They have conformational changes. So there are in amino acids, you know that there are side chains, and so in protein structures there are parts which are very flexible and little parts which is not really flexible, and so they are extremely plastic, so they can adapt different shapes, and usually these molecules are globular and stiff or elastic, and they change conformation according to their function, or when the activity occurs, it can change the shape. As I said earlier, they are machines synthesizing small or large molecules and catalyze their breakdown. Like enzymes work on like catalysis, so they do like synthesis of different molecules. So I have just shown a couple of figures showing like ATP synthase motor domain. Like there's a huge one there at the end. The second one is Manduca sexta. It's insect. This insect chitinase. The other one is GBP, glucose galactose binding protein, which you know that one. Then the fibrous proteins. Okay, so the organization of protein structure, we say there are like 4 different levels we usually discuss. It's primary structure, secondary structure, tertiary structure, and quaternary structure. So it's the primary structure is the amino acid sequence. This first just letters. There's no sense into that when you don't know, but it is a protein gene. No, it's a protein structure. It's called primary structure. It describes the amino acid sequence of the protein. So there are like 20 amino acids, standard amino acids, and there are different properties of these amino acids. So based on the properties, this organized molecule have a different function or different properties. So then the protein is made up with peptide bonds. So 2 amino acids form a peptide bond here. I did something wrong. Okay. So here's the peptide bond. So this part is called main chain. So it can grow, making another peptide bond here, and it can grow like that. So it's called a main chain. So here I have shown a main chain. So it's organized. These are side chains of different amino acids, and this is the main chain of the protein. But although it looks like this in the figure, when it forms, it folds into an active protein. So this when the protein folds, inside the protein molecule there are local structures which looks like an helix. So they are called alpha helix. So it looks like they looks like this, and there are bonds making it like a cone. Cylinder. So these parts are called alpha helices. Then in the protein structure, it also has another arrangement called it's this amino acids arranged like a zigzag in a zigzag manner, and these are called beta strands. So this is a beta strand, this is a beta strand, this is a beta strand. When they sit together, they make interaction with each other, and it's called a beta sheet. So because it looks like a little sheet type structure. Then, after the protein is folded and it makes a functional molecule, it has a tertiary structure. For example, these three. This is binding protein. I will come to this later on. It's called pili protein, fimbrial adenosine, and this is an enzyme which is cell 7. It's a cellulase, cellobiohydrolase from Trichoderma reesii, and this is salmonella glucose galactose binding protein. So, if several or couple of these proteins get together and form another structure, another functional unit, it is called a quaternary structure. So, for example, this one is Rubisco. These different colors shows different tertiary structures. So, it is a huge assembly. The function of it is to trap carbon dioxide in photosynthesis. And this one is a virus. So you can see the virus structure, and it has, I don't know how many proteins in this one. It's a bacteriophage PP7 from Pseudomonas aeruginosa. So this virus attack Pseudomonas. So to have a structure, we need proteins, right? I try to speak slowly because I want you to understand what I am saying, otherwise it's useless. To determine a protein structure, you need to have protein, right? So how we produce proteins? Now there are many methods of doing it, like some fungi, for example, or bacteria, they produce proteins and they secrete it outside. So you can collect that. Or once I work on a project project on chitinase from yam. It's a special sort of yam produced in Japan. So they bought like kilos of yam and they mash it and purified the protein in the lab. So it's the other way of doing it. And now we need constant supply of protein. So what we do, we clone the gene. So we take the gene, we can buy it now, we can synthesize. If you know the amino acid DNA sequence or gene sequence, we can get this help from a chemical synthesis company. We can buy it, the gene. We don't have to do all the lab work from the bacteria and isolating the gene. We can do that also. So buy it, and then we can clone it into a vector. Vector is clone it into a plasmid. Plasmid is a circular DNA. So we can insert the gene into that, and then we can produce it. So we use different other helping things like electrophoresis. We want to see whether we have cloned it or not, whether the protein is expressed or not. So we use something called electrophoresis to understand that. So then you have to know a little bit of, not little actually, you have to know a lot of microbiology. Then because you have to grow this, first you clone it into the plasmid, and then you insert it into we call this transformation. We transform it to a bacteria, usually E. coli. There are other many systems like yeast. You can use yeast, Pichia pastoris, Saccharomyces cerevisiae, or there are several other fungal strains as well as bacterial strains. So once you insert this plasmid into the bacterium or fungi. Then you need to grow it, and you isolate one colony from this sort of plate, because we expect that gene is there. And then, of course, we do a lot of quality controlling before proceeding, because we have to know whether exactly we have inserted the correct gene there. After that, we can express the protein. So there are like promoters, that means different parts in the plasmid. It's telling the gene to express. Okay, now it gives a signal. Okay, now you express it. So then we can express the protein. We can do one is in the lab. We can use something called shake flask cultures. You grow it in a flask, in a shaker. And you can, there are a lot of properties to understand here, like temperature, and there are several other factors affecting on this protein expression. And then, or you can use this kind of fermenter. These are industrial things, like when you want to grow like a lot of protein, like I think in this one you can grow like 15 liters at the same time. So it's once you grow the bacterium, I forgot to mention one thing. Sometimes this protein, the gene which you express, it direct your protein into the periplasm, a special cavity or place in the bacterium between the outer wall and the cell membrane. Right, that's one thing. Or there are strains, it directly secrete the protein to outside. Based on this, so if it is expressing into the periplasm, then we need the bacteria. We have to separate bacteria and the broth, right? So in order to do that, you can use either centrifugation. You put it into a bottle and you centrifuge it, and then bacteria settle in the bottom, and then you can, if it is in the broth, then we can take it, or if it is in the cells, then we proceed with that. Or you can use something called this ultrafiltration or microfiltration. Actually, microfiltration is to separate bacteria and the medium, and ultrafiltration is protein. It separates proteins. It's a next step. There are machines and they are very expensive. That's one of the things which we can't do many things in our country. So here, then we proceed after centrifugation. If we have the protein in the cells, in the bacterium periplasm, we use one of these. These are called cell disruptors, or there is another name called French press. Maybe different companies use different names, so you know. So, but what we do here, you suspend the cells and you put it into this little hole here in this container, and then it sends with a high pressure in a small hole. There's a small hole inside that, which the pressure is very high, so all the bacterium, they burst and all the proteins come out. So it's a mixture of protein, cell debris, and all sort of contents from the cells. So and especially DNA, which is a very difficult thing here. So you add DNAs to destroy DNA, because you can't proceed after that. Otherwise, very difficult. So then there are other methods to extract proteins. You have enzyme digestions or sonication. There are you give a high frequency wave to break the cells open. So there are methods of doing that. And then you centrifuge it, all the thing, and then you get the extract, which is with your protein, your interested protein here, right? So briefly, I just explained you about like a whole PhD project. Then what we do next is we have to purify the protein. We have to get the protein out from that whole mixture. So there are techniques. So in these proteins, or these molecules, have different properties. They have charges. They can be, you can manipulate these charges, or there are patches which has like a hydrophobic patches, or maybe these molecules are have a like affinity to something. Then we can use that property. So before going there, you have to analyze, you have to read about it, you have to analyze it. We can do a lot of things like using bioinformatic analysis also. We are going to do like models, and then you understand the protein first before you do this actually. So then we use these different properties to separate these molecules, right? So we can use like hydrophobic interaction chromatography, affinity chromatography, ion exchange like cation exchange chromatography, anion exchange chromatography. Like there are a lot of different techniques to do that. At the same time, people use tags. Then when you clone your protein, you put a tag at the end. Like say if you want to catch a ship, you attach a boat to it, and you can catch the boat. Then you already, it's automatically you catch the ship. It's something like that. So it's a big protein, and you add a little tag, and you use this little tag to purify proteins, because when you have, like, for example, there is a tag called histidine tag. There, you can add some 6 histidine amino acids, like 6 or 8 or 12, whatever, and then it has an affinity to ions like nickel or cobalt. So then you can use that to purify the protein. So once you purify the protein, these are called ACTA machines, ACTA purifier. This was now made by Cytiva, a company in US and especially in Sweden, but owned by a US company. So there are various columns with resins having different properties. So you purify the protein, as I said, and then you can analyze it. Did I purify my interested protein or not? How you know that? So we have to analyze it, we have to understand it. So there are a lot of, or several biophysical, biochemical methods to analyze your protein. So that's why I have just listed like few, like HPLC you can use even to measure the concentration of your protein and the purity of your protein, and then ITC to understand the interactions with the different molecules like ligand protein interaction or protein protein interaction, and then DLS you can this dynamic light scattering, so you can use this for similar purposes like this interaction analysis, as well as to see whether you have no aggregates in your protein preparation. It's thermophoresis, microscale thermophoresis. It's also used for analysis like interaction analysis. Then SDS page is sodium dodecysulfate polyacrylamide gel electrophoresis. It's a electrophoresis. This is used for protein analysis. Earlier I mentioned electrophoresis. There is something called agarose gel electrophoresis, which is for the DNA, RNA, and nucleotides, right? So then you can use something called thermal shift assay to understand the melting temperatures of proteins. If a protein is bind to something, say this is a protein and this is a ligand, it binds like that. And if you melt this, it takes a high it needs more energy to to release this, or to degrade this, or to denature this. But if you have a protein like that, to denature this, it's not that much energy you need. So using this, you can make it like a, you can calculate something called melting temperature, right? So if something is bound to the protein, then the melting temperature is high. So using this kind of technique, you can convert that into different signals, and you can measure it, and then you can understand your protein. So there are a lot of other spectrometric analysis methods, like to for example, to measure concentration, or if you do assays to understand the protein activity, for example. So of course you can use statistics, or sometimes in a big companies, in large scale procedures, you use something called design of experiments. It's a statistical analysis method which you use to understand the production of proteins. Why this is important? Think about, you are using now there are something called biologics, it's a protein drugs. So if you want to make it like in bulk, you need to grow like you have to have a big fermenters, and you need liters like 2, 3, 500, or there are like 1,000 liters fermenters, even more actually. There are bigger ones. You need, of course, like forklifts and stuff like that to lift these big fermenters. So the drug in the drug business, it is very important. So that's why I mention all these. So once you have the protein, then we can, now you have the protein, now we can try to understand the protein in a different level, like a structurally. So there are 4 usual widely accepted methods to understand the protein structure. One is called X-ray crystallography, which is the mostly or widely used method in the world right now. And there's another one called NMR spectroscopy. So there are some problems with that, because you cannot use this method for the large proteins. It's only for small proteins. The not novel actually, now emerging sort of method, it's the people are trying to use a lot, is electron microscopy. It's called cryo-electron microscopy, because it's handled in under like very cold conditions, like -100 maybe, something like that. So it's called cryo-electron microscopy. And modeling is just fantasy, right? It's nothing to do with but you can understand a lot about structures by doing that. It's a computational methods. Okay, now structure determination by X-ray crystallography. So to do this, you have to have a pure protein. So at least it has to be like 97% pure. Otherwise it doesn't, if there are impurities, it will not work. So once you have the pure protein, then you need to make a crystal of the protein. You can, if you have a salt solution or a sugar solution, you add take water and you add salt, dissolve it. And there's a point which is you cannot dissolve any salt more. That is called saturation, right? Because it is saturated solution. Once the saturation is like super saturated, then it starts crystallizing again. This is the same technique they use for, like, if you do it for sugar, then you can make this, we use it like rock candy. It's called sukiri, yeah. So it's the same technique you use to produce that. So we can use that technique to produce protein crystals. So there are many different solutions, like polyethylene glycols. There are different sizes and various chemicals. Then we can add into the solutions and add protein into that and keep it there for some time until the crystals appear. So crystallization is a natural process. So we can just help put in one and one together, and we cannot make crystals. So it is a natural thing. So so this I am talking about very tiny volumes here, because we don't have kilograms of proteins, right? We have like say 10 milligrams. We have to do like hundreds or thousands of experiments with that. So and the concentration of the protein solution has to be very high. So what we do, we use smaller volumes. Okay, so if 1 microliter means 1 milliliter means 1000 microliters, right? 1 microliter means 1000 nanoliters. So this kind of robots, these are pipetting robots, they can pipette like 50 nanoliters. So we can do a lot of experiments with a few microliters of protein. So this is also a robot which is developed by something called Douglas Instruments in the Texas, in the United States, and this is a British company produced this one. So once we have the crystals, then we put it into a little loop. So the crystal size is maybe like, I don't know, it's micrometers, like 0.1 micrometer, 0.05 micrometer, 0.2 micrometer. 0.2 micrometer crystal means it's a huge crystal for us. So we have to manipulate all this under the microscope, and then we pick it up, and then we flash freeze in liquid nitrogen, and then we put it into a special container which is called a dewar, and then we bring it to a X-ray source. Okay. Sorry. So we bring it to X-ray source. So here is a, for example, X-ray source. I will go into this a little bit in detail later. And then in a X-ray source, there's a special experimental stations. So you can put this crystal into an X-ray beam, and then you can collect the X-ray diffraction pattern from there. So this is the X-ray diffraction pattern, and then you using these patterns, you can calculate electron density, and then you can solve the structure finally. So these are different crystal forms and different crystals. So they are in our, like, it's like 2.5 micrometers. These are very small crystals, yeah. Different crystal types and different proteins. They crystallize in different forms. So, as I said, X-ray, then you put your crystal in the middle, and then it give a diffraction pattern. Based on this diffraction pattern, we can calculate the structure. So there are two different X-ray sources. One is home source. It's a machine which is like a usual room. It's a, the equipment is that big. And the other one is called synchrotron. It's a huge lab which produces X-rays, intensive X-rays. Okay, the synchrotrons. This is a very interesting lab. There are many in the world, I think about 70, but I don't think all we can use for protein crystallography. We can't use for protein, but they use for different other material science and other chemistry research. So what happens here? Okay, so here I have shown several, this is called European Synchrotron Radiation Facility in France, this one in Grenoble, France. This is called Max 4 Laboratory, it's in Sweden. And this one is Spring It. It's in Japan. You see, this is built around surrounding this little hill. This is it's more than 1 kilometer. This is exactly like 1 kilometer, and this lab is more than 1 kilometer, and maybe 1 and 2.2 kilometers or so. This is rather small but a very powerful synchrotron. It's called Diamond Light Source. It's in close to Oxford, and this is another synchrotron in Japan. It's underground, and it is called Photon Factory. It's one of the first synchrotrons. So this is Daisy. It's in Hamburg. You see, it's I think it's about 4 kilometers long. I think it's underground one. But here, all these, this one everywhere there are laboratories and beamlines. But here it's not. It's only this part is the lab in Hamburg. It's oh, okay. Oh, thanks. Okay, in a synchrotron, what happens? So it has electron gun, right? It's called LINAC, or linear accelerator. So you take some electrons. By there, I have listed here how they obtain electrons. Say, think about that. You just get some electrons. Then you put it into this accelerator and you speed it up into light speed. And then you inject that into something called storage ring. You saw the earlier figure, this round laboratory. It's inside that there is a storage ring. It's a vacuum ring. Actually, it's not a ring. It's made of like straight stretchers. It's a polygon. So it's like that. There are always bends between these stretchers. So using something called bending magnets, they bend the electron flow. So it's a vacuum. The electrons move in light speed. So in these bending magnets, at that point, it produces X-rays. It's not that intensive, however. But there are something called insertion device. In the middle of the straight stretchers, you can have a special sort of magnetic, it's like not a magnet, it's a magnetic system. It's a huge area. So, due to these different poles in the magnets, like in here, and it produces X-rays. So this X-ray, in this kind of thing, it produces very intensive X-ray, extremely intensive X-ray. So this X-ray, like a parasite, we use it for protein crystallography. So this is an experimental station. Now this is a little bit old, actually. Now the new ones are maybe different, and it's depending on the synchrotron to synchrotron, experimental station to station is different. But here, so X-rays come from there, like there's a little pipe-like thing here, and this is a detector. So you put your crystals there. So this is just a robot mounting the samples, because samples are dipped in liquid nitrogen, right? So the liquid nitrogen is minus 196 degrees centigrade, and here with this one, I think it is the one. It's not clear at all. Yeah, that one. You pump liquid nitrogen into onto the crystal. So then we can maintain at least minus 100 around the crystal, because this intensive X-rays otherwise burn down the protein crystal. So this kind of setup we use for data collection. So it's a close-up of that, not exactly that, but it's another system. So you put your crystal here, X-rays come from there. This is for the liquid nitrogen or the gassed liquid, liquid nitrogen in a gas form, later on a gassed, but it is like minus 100 temperature here to maintain. And there are a lot of cameras and other sensors and a lot of things in this experimental station. So here I have shown here how a sample mount. So there is the loop is sitting at the end of here. So and we just put it into the X-ray beam. We can adjust it to do that, to exact, because we are talking about very small area of X-ray beam. It's like 50 micron to 50 micron or something like that. It's a very small beam. So then there are detectors. So the earlier, the ones which I showed you, they are the earlier detectors. But this is the newest generation of detectors. They are called pixel detectors. So you can measure each pixel of the diffraction. So you can get a much better idea about the structure using these ones. This is a sort of old detector. So X-rays are, you record the X-ray diffraction here. But the long time ago, like in 80s or so, they have used photo films, like huge films, to do that. Not like this. And they had to calculate by hand. The computers were not available, and so it was tedious job at that time. So this is a usual X-ray diffraction pattern. So using these dots here, we can calculate the electron density of the molecule. And of course you do like a lot of averaging, and there are a lot of calculations, so I am not going into the details of any of those, because it's too complicated for you to understand in this kind of seminar. So then we have powerful computational methods to solve the structure, so to calculate the electron density. And then once we calculate the electron density, we fit a model into that, and then we do something called refinement. We can heat computationally, we can heat each atom, say to 5,000 degrees, and then we let it settle inside in the energy minimized place within the electron density. By doing that, we can at least get closer to the real structure. That's how we do the refinement, and then we get the final structure. So this is just electron density, and electron density, this is in the blue, and then this is an amino acid, this is called tyrosine, and tryptophan. So I have just showed it, and this is a catalytic site of one enzyme. I think it's this is glutamine, aspartic acid, and glutamine there. So this is the active site of one enzyme, which is called cellulobiohydrolase from Phanerochaete chrysosporium. It's a fungal strain, white rot fungi. So I just wanted, for the teaching purposes, I have just made it. So the side chains, which I showed you in earlier figure, they should have this kind of like electron density, you see. Of course, there are much better electron density than this in some maps. It's depending on the map quality and the resolution of the structure. So I just wanted to show you how it looks like in the electron density, because everything should go inside the electron density. That's the only thing we can trust. Then we can trust it, because it's experimental results. Once you solve a structure, it looks like that. You don't see much details, right, in this case, because but then you have to go into a computer and go into details of the structure. This is like a bush, right? So it's very difficult to understand, tedious to look at it, of course, other than a structural biologist. I mean, if you are a structural biologist, even I cannot just see anything here, because, of course, this one I know, but if I see for the first time, I don't see just some amino acids, that's all, because I can't see any functionality in that one. So we have different representations, right, to show different things to the people. So here is the normal wire mesh, normal ball and stick model which I showed you, and this is called surface calculation, and this is the same thing with another view and another, like it's called net or mesh type show representation. This is the normal thing we showed it. It's called some people call it a cartoon figure, and this is called space filling, and this is the main chain. I have shown it here, the main chain of the protein, no side chain there, because these different things are important to explain different properties and different characteristics of the structure. Okay, so what can we obtain from a structure? Now you saw things, structures, so you can get the shape of the molecule, right? If there is a tunnel, if there is a cleft, if there's like, you can see that. And then you can obtain catalytic site, substrate binding site, product binding site. Now I am talking about an enzyme, right? So these are available, they are available in those enzymes. And from looking at it, we can obtain the catalytic mechanism or binding mechanism or the other sort of interactions. If there are cofactors bound to the protein, like ATP or whatever, some other molecule, or ions, usually some enzymes or some proteins need manganese ions bound to the protein or magnesium bound to the protein in order to act. So we can figure it out from there, and then you get functional insights how it works. And then if you want to improve this protein for some reason, then you can understand where to improve, how to improve, and things like that. So I have just shown some figures here. So you see now this is a cellulase, and so the substrate is coming from there. There's a tunnel like that. So it goes inside the tunnel, and at the catalytic site it breaks into cellobiose. It's a product, two sugar units, glucose units, and it leaves from there here, like this is cellobiose, so it leaves from there. So there's space for that. Here you see it's easy to leave from there. So now some examples. Okay, so this is phanerocrite chrysosporium cellobiohydrolase. So I have done a lot of like extensive research on this molecule, like to see to obtain each conclusion or the idea, you have to see it in the structure, otherwise you can't do that. So when we saw the structure, we do it like nothing bound to it, and then different molecules bound to it, and then we put everything together and try to understand whether there are changes in the structure or during the binding or affinity to any other molecules, etc., etc. So always we have to show, or we try to show if I put it in like this, say to prove that this is the product or this is bound, we have to show it with electron density. Otherwise, you can't trust. Who knows, right? It's you can model anything in the space. So that's why we show it like that. Okay, I have fit the correct molecule into the electron density, right? So there also. So based on that, we can do activity assays and just show that the bound molecules really inhibit the protein. It's very important to understand the inhibition, because if you go for, if you want to stop inhibition, you have to understand that, and if you want to make a drug for something, some protein, not in this case of course, but if your interested protein is a drug candidate or drug target, then you have to know exactly. Inhibition, you can do biochemical assays, biophysical assays, and then by doing this, both putting all this information together with the structure, you can understand the catalytic mechanism, exactly how it works. Like in this case, like how it breaks the cellulose chain, how it makes like this boat conformation in this one, and then it breaks, and then you see here the position is this is a equatorial position in this bond is retained here, so it's called retaining mechanism in this cell biohydrolase. So this is called glycoside hydrolases. These enzymes are widely known as like that. So I just put this into a little cartoon, so how it goes into the enzyme and it breaks there and it leaves from here. When I made this little clip in, I think it was like, I don't know, about 20 years ago, the technology was not so good, so we had to, I had to work like 3 weeks or so to put this together. So it's a completely handmade thing. So now it's easier to do this sort of thing with graphics and the development with computational systems. Okay, so now we talk about what we are talking about, molecular machines. So I have to prove you that it works like a machine, right? So that's what I was just trying to do. So when there's a cellulose chain, there are enzymes which breaks from, they are called exoenzymes, from the ends. So this is reducing end of the sugar chain. So some enzymes work from there, like from this end. Some enzymes work from the other end, non-reducing end of the sugar chain. Some enzymes work from the middle. So the enzymes which, the cellobiohydrolases, the enzymes work from the ends, this goes inside and they start cutting like that. And so it happens in both sides. But the enzymes working from the middle, they sit on the middle and break this sugar chain into two, right? And then it sits there, breaks, sits there and breaks. So it's a huge system, because it's same fungi should be able to degrade wood and eat. That's what they do, right? These are fungal enzymes. So this is the product of this is cellobiose, and product of that is also cellobiose. So endoglucanases, it's the acting from the middle of the sugar chain. They produces various sized oligosaccharides. So this enzyme called beta glucosidase, they eat it and they produce glucose. So you take a wood, use all these enzymes, and you can make glucose. Why we need glucose? In 1990s, there was like huge money from the European and US government to produce ethanol, lot of ethanol, to use it as a fuel. So in Sweden, we have ethanol buses, like public transportation is run by ethanol, using ethanol. And in the US and of course in Brazil, they don't use this, but they use molasses from their sugarcane plants to produce ethanol and run vehicles. So there is a, we can use that, and there are some companies produce cars to run by petrol and ethanol. And in Sweden, now the petrol here is octane 92 and 95. We have 95 and 99. So in 95, the 95 is octane, 5 is ethanol for us. And there's another fuel called, it's called E85, which is 85% ethanol, 15% octane. So that's why we need this sugar. That's why they put a lot of money into these products, these projects, to understand all these enzymes and to use these for the production of sugar to ferment and to produce ethanol. So in the US they use a lot of the corn stalks and hay, for example, to produce ethanol like this. Of course, there are other chemicals in this, so you have to understand how to remove it, and there are a lot of other like hemicellulases and different other enzymes to get done that sort of thing. So that's why this is very important. Next, chitin active enzymes. So this is the second abundant biopolymer on Earth, chitin. It's shrimp shells are made of chitin, right? Shrimp, and then there's squid. They have little hard part in squids. Those are also made by chitin. Chitin is it is N-acetyl glucosamine. So there is an acetyl group. Here, if you remove the acetyl group, it becomes something called chitosan. Chitosan is soluble in acidic medium, and there are a lot of products produced by using chitosan. It's like burn wound healing plasters, for example, and creams, facial creams, body washers, deodorant sticks, and using this material. So the chitosan, this is also very interesting because especially in Asia, not in Europe, and they like to probably have the product used, but the basic research is done in like Thailand and East Asian countries. So chitosan is very interesting. So the enzymes, there are a lot of enzymes working on chitosan as well. So I have just mentioned few like various chitinases and chitin deacetylase to produce chitosan. So chitinases, there are different chitinases like earlier as I showed you this Manduca sexta chitinase, and this is Bombyx mori. It's also sort of beetle, I think. Then plant chitinases, there are chitinases in plants. So this is a very interesting research area. Why plant have chitinases? It's also interesting because you see now we have an immune system. Plant, they don't have anything. So how they prevent attacks? So once fungi attack them, fungi cell wall has chitin. So what plants do? They produce locally. They produce several molecules, including chitinases. So the plant can attack the fungi again, and then they make it a little knot-like thing, and it's separate from the tree. So that's how plant take care of that. So chitinases are it belongs to a group of proteins called antimicrobial peptides, because they work against antimicrobial microbial attacks. So here it's one. This is from Brassica juncea. It's called Chinese cabbage, I think, or some sort of salad lettuce used in like Eastern Asia mainly. So I'm pretty sure in mustard also the same, because this is similar to mustard. It's very close relative of mustard. So here, once we solve the structure, we can see it's it's bilobed, right? It has like huge cleft, like here. You see? So this is the information I could get, because I couldn't get anything bound to that because it has a huge cleft. So, but we could understand that it has a mechanism. Earlier, I guess you remember the name retaining mechanism. But here, in this case, it has something called inverting mechanism. It's the orientation of the axial position of the OH there, or O, between the sugar molecules and this becomes equatorial position becomes a axial position in this one. So it's it has like a long distance between the catalytic residues, so that's why we guessed it like that. Okay, we couldn't do much because we didn't have enough information. And we also observe, when I saw different structures with different conditions, I observed these sort of movements here, like a little movement there, like large movement there, and so some here, for example. So this is how we say protein has conformational changes during the activity. Otherwise we can't. We don't have proof. We can speculate. In science, speculation is, it's something else. You have to prove this happens. If you want to say something, you say straight like this. It has conformational changes because you prove it with scientific evidence. So in order to do that, I had to I showed everywhere like this, in these moving parts, the electron density in the, so that's how we prove. So this is just to show how much movements in the different molecules. So when the substrate comes, it closes here and it holds like this, the substrate, and then it eat it, right? So when I compare these chitinases, this brassica juncea chitinase, it's a mustard chitinase, with two bacterial chitinases, I could see there are some loops missing, like it is there, this is there in a different conformation, this is missing. Oh, in plant one, in brassica we have this, in bacterial one we don't have that. Here in bacteria we don't have this. Here in bacteria we don't have this part of the protein. What does that mean? These parts are not so important, right? That's why bacteria, that's the evolution. So when things evolve, it's, they do these sort of changes based on whatever the reasons they have. Then I worked on another protein called Norway spruce chitinases. So this one, when I used that one with brassica, so here this is missing in the Norway spruce, this loop part. This is not there. This is not there. So, but in all the structures, all these chitinases, this is called family 19 chitinases, and all these structures, they have this part. So from this, what can we conclude? We can, there's another thing. With this one, you can, there's another papaya chitinase. So when I put this papaya and mustard together, we can see this papaya is more close than the mustard one. This means when the substrate, all this information, this, to solve a structure, it takes months to do the crystallization, and it's a lot of work, but the conclusion is very little. So what we can see from all these structures is that when the substrate comes, it holds it like that. Yeah, there's a special one loop which I showed you. It's enough that the substrate comes, it hold it and it eat it, breaks it. So this is how we have to prove scientific evidence. It takes a long time, but there are no shortcuts. So these some of these chitinases are multi-domain enzymes. So there is a catalytic domain, and there's a little connection, and there's another domain which is we have identified some of these as substrate binding domains. So you see, it's very easy, right? If there's a protein, so it can hold into the substrate and then it start breaking it. Otherwise, if it will be just flowing around in the water or whatever the solution it is there, even in the plant system, plant within a plant or in a bacterial system. So substrate binding domains are very important. You can just understand this by reading or understanding that. But how to prove it? That's another thing. So this is my speculation. I thought, okay, this is Norway spruce, okay. So and it has this, and it has two domains, two substrate binding domains, and it binds to chitin in fungal cell wall, and so it can break chitin like that. So I did a little test. So I make a little agar plate and make 3 holes in it, and I put this Heterobasidium annosum grown piece of gel here, and then I put this in the A hole. I have put this is Norway spruce chitinase with chitin binding domain plus catalytic domain, and in the B it's only the catalytic domain. It's just only one blob, right? And the other one just buffer, and see what has happened. So if the catalytic domain alone can slow down, break the fungal cell wall, then it does, but it is not effective. Here with the substrate binding domain, it has completely inhibited the growth. So that's the importance of the substrate binding domain. So I did another test with that. I had some chitin flakes which sent by a company to me. So I just put the enzymes and incubate it for 24 hours, and then I observed under a scanning electron microscope. You have to have it something like that to magnify 10,000, 20,000 times. So here this is just chitin, and this is chitin with the chitin enzyme catalytic domain alone, and this is with the substrate binding catalytic domain fused to substrate chitin binding domain, and so see the difference. It can break, and this is a small enzyme is a very small molecule, but chitin is a thick just piece of shrimp. See the difference. Yeah. Okay, let's move to the next. So I just speculate my theory with this little movie clip. Okay, so let's. I just want to briefly mention that in this bacteria, they have hairs. These hairs consist of proteins. They are complete protein structures. So in this particular one, it has many different. At the end of these hairs, the molecule the molecular part is called adhesin, because it interacts with the epithelium of urinary tract, and it infects the urinary tract. Especially for the bedridden elderly people, if they have this bacteria, it's very difficult to control. There's no way of, it doesn't, because think about it, I mean, this little thing attached to the cell wall, and urine is passing in the urinary tract. It's like a huge river, and this is like a little something, hang on to the wall. So how strong it has to be to sit there? This is very strong interaction. And so in these ones, I have worked with some of these adhesins. So this is one of those. It's called MRPH, and we don't know what is the interacting point in the epithelium. But the idea of this is if we can develop a drug which goes in the urinary tract, so it can bind to this the hair of the bacteria and stop interaction with the epithelium. Okay, there are another example. It's a periplasmic binding proteins. There are proteins which used to transport different molecules across the cell plasma membrane in bacteria and gram-negative, gram-positive. It can be any bacterium or any cell, even in humans. So we worked on glucose galactose binding protein and from salmonella. So this is the natural substrate of this binding protein. So what happens? It binds the molecule there, and then you can see the different conformational changes. What happens? It is like that. So when the sugar binds there, it close it and bring it wherever, and it releases. So in order to prove this, we have to have, like, this is, I think, 20, 30, 40 structures. We have we have done a lot of experiments to prove this. So this is another protein called xylose binding protein. So we fortunately we were able to get the, you see here it's ligand, it's called ligand free form. So there's no ligand here. So now it's ligand is xylose, and then after it's just, this is the starting of the binding, and then this is the end of the binding. It's bound and closed, and then now it transport into the cell plasma membrane, and then it releases there. It's like this. So there are free forms, then bound forms, and bound open forms, and then it brings here, and then it releases to this pump, and it the transport protein, and it transport inside the cell, right? Okay, so I was just giving you examples and protein crystallographically determined structures. So there is another method called homology modeling. So if there's a structure, and if there's another unknown protein, but which is very similar, structurally similar to that one, that means identity of the amino acid sequence should be more than 40%, then we can make a model to explain that protein. If you don't have experimental tools, if you don't have any other way to know this, homology modeling is the best way to, get some information. So I have done this a lot for many proteins. So this is just a list of things. So we understand, like this one, this synthetic antimicrobial peptide is a very small peptide. This is a modification. When you have a wound, body produces this thing. And of course, not exactly this. It's a little 3 amino acid shorter than this, and in the body, which is called L37, I think, as I remember. And so it is the one has antimicrobial properties. So that's why it prevents the bacteria attacking to our wounds. So we, a group in Spain, they contacted me to work on this, and they modify this, and then they use it in the fields to control pathogenic control, I think, in cultivation. I don't know which one. So we can use homology modeling, and this is one of the models I use. They put it into this book cover. So by homology modeling, we can understand a lot of things: the structure, the way, the catalytic mechanism. For example, the phenorochete, which I showed you, it has 6 enzymes, isozymes. Why for the same purpose it has 6? It's useless, right? So, but the nature is clever, so there should be some reason. So we want to understand that. So I modeled them. That's the only tool which we have. And then from like 2 of these, we understood there are drastic differences, including here this one. It's called one of the isozymes. It has even a different catalytic mechanism. Like there are changes in the catalytic site. Usually, catalytic site is conserved in all the enzymes, similar enzymes. If it is changed, that means it's a completely different enzyme. So when we don't have structures and it's costly, take long time, lot of money, so modeling is the option. So we use this to understand molecular evolution of these chitinases in Trichodoma species. So it's for the mycoparasitic interactions. And another example, Streptomyces coelicolor, FTSZ. So what happens? So it's like a tube-like thing. So when it wants to divide, bacterial cell division, they make a ring structure around it, and then from there it makes the division. So this protein is involved in that, and I think there was a Nobel Prize also for the bacterial cell division. So we didn't have the protein, so we used homology modeling to understand that. Say a protein structure is a chair. So if you know the chair, okay, you know what to do, why it is happening, or what is the function of it, for example. So think protein molecule as a chair. So in the case of chair, the purpose of this is to sit, right? So if you want someone, preventing someone sitting there, what you can do? You can just put something on there, right? So then you can't sit. This is exactly the same thing we do in drug designing. We find something very important to us, for a cancer, not important to us, but for the cancer, and for the bacterium or whatever the organism we are aiming at, and then we find a molecule, a protein molecule of that organism, or in a cancer, for example, and then we design another molecule, another small molecule, to block the activity of that. So that is a drug, in simple terms. So this is the same thing. In this case, this is para-space, so there is a catalytic site, so you can design molecules to block the activity here, as well as there is a allosteric site, so you can design molecules to sit here to block the activity of this para-space. This is very important for I worked with this for some time to develop drugs for bladder cancer. So I cannot talk more about it because it's this company secrets. And then if we want to improve, like industrial enzymes in the ethanol production, for example, so we need to improve enzymes. We cannot trust completely on the natural enzymes. So we can understand when it's like the chair, right? When we know what to do with that, then we know what to improve and what to do, modifications. Because like for the enzymes, we need enzymes with high activity and work in high temperature, have a very fast activity rate and large pH range. Like say if you want to, if you can have a cellulase acting in 50 degrees, then you can use it in a washing machine, right? In a washing powder. Then it remove all the dirts and breaks the cellulose chains in the cloth and make it nicer. So, and we need a stable protein as well. So in order to do this, we have to understand the structure. So how we do this? Then we understand what we have to change, and then we genetically modify the gene. So like site-directed mutagenesis, for example, so you can change one amino acid or several to get, it's of course trial and error. You do it and then you test it and then you understand it. Otherwise you do it again with another set of ideas. And also you can have different enzymes in different organisms. So you take an enzyme from one candidate, or say one fungi, and put it into another and make it much better. So there are many ways of doing that, enzyme engineering. So to explain it, say if you want to make this chair stable, so for example, you can just put something here, right? Like a screw here. Then it makes more stability. So it's like a one amino acid change. It's like site-directed mutagenesis. You can do it in protein. And say if you want to stabilize the legs, you can, for a chair it's easy, right? But for the enzyme, you can do the same, or enzyme or whatever the protein. You can introduce a disulfide bridge, disulfide bond like two cysteines, and then it makes a bond which is making a stable enzyme. If you want, say, now there's another thing. If you want to increase the rate of sitting on this chair, what can you do? Like say with this kind of chair, maybe like two people can sit within 1 minute, right? One comes, sit there. Then what if you modify this like that? Then maybe you can, like, 4 people can sit in a minute, right? This is practical. So if you do like this, maybe 10 people or more can sit. But this is how we increase the activity of an enzyme. We do changes, we take away parts, we introduce parts, we can change a lot of things in doing genetic engineering and produce the enzyme with our modifications. So because I think this is easier to explain, that's why I use this chair thing. So Trichoderma reesii cellulases, a company actually, is a company did this, all this with a set of professors in my previous department, Agriculture University in Swedish University of Agricultural Sciences. This guy is my colleague. We shared the office room when we were PhD students. So they did a lot of changes, and they could find, you see, this is the change in melting temperature. So this has increased, like this is almost 11 degrees. This is like 10.5 degrees centigrade. So say the optimum temperature was earlier, say 50. Now you can use it like in 60 without any problem, because this product, this enzyme is used in ethanol production as well as in washing powders. So this is my last slide. The idea of engineering of these kind of enzymes is to have an stable enzyme to use in an industrial system. So if it is unstable enzyme, with the application time, you see this activity is, it reduces, the activity reduces with the time. But when you make an stable enzyme, you can keep the activity in the same sort of strength or the pattern. So this is what we usually aim for in the industrial developments. So to conclude, we can say that protein structure, the proteins are like machines in any living system, right? And to understand that, we need to have the structure. So we can solve the structure using different methods, including protein crystallography, or if you don't have, like in a condition in our country, in Sri Lanka, we can use homology modeling. So it's what we need is the positive thinking attitude to do all these things that we can do. So I wish you good luck, and I would happy to answer any questions you may have. If you want to ask me question in Sinhalese, no problem. I don't understand Tamil, unfortunately. So, but yeah. Yeah. The question is how we get crystals if we have ligands or inhibitors and the protein. There are no easy ways of doing that, but first, the easiest is that if someone has crystallized the same thing, then it's easier to do that. If it is a new one, then we have to do something called screening. There are screening kits with different solutions, like they are expensive, of course. We can use those kits to understand, or you can handmade with different components. But handmade is a difficult thing because you don't have the same all the chemicals. That's another thing. So the chemicals has to be very pure, whatever the chemical you use, and then you have to first screen for crystallization conditions. If you see some signs of crystallization, then you can optimize that condition. You see, like there are signs like you get aggregates and needles which you can't use for experiment. But if you get a needle, then you can crush it. Then you can use that as a seed to generate crystals, and then you can do the same thing again, and then you probably get a better crystal. Or you can you take the same same condition, and you can change little bit of that. Let's say if it has polyethylene glycol 33, 50, 20%, for example, then I may change the polyethylene glycol concentration like 16, 17, 18, 19, 20, 21, 22, 23, 24, like that, and use keep the other parameters constant, the same buffer condition, same pH, for example. And or you can change the pH a little bit, you keeping the other conditions constant. By doing that, you can get a crystal. But get crystals, sometimes crystallization is it can take days or weeks, so we don't know yet, and sometimes we don't get crystals. So because if the protein is glycosylated, you see, in nature, I told you that there are protein with several domains, so one connected to another one. So in nature, what happens, these proteases breaks these links. There are attacks everywhere. So in the molecular level also, there are a lot of attacks for the other molecules. It's like our society. So in order to avoid these sort of attacks, what the nature has done, they have glycosylated these linking parts. That means glycosylation is adding sugars. So there are sugar chains, like branches everywhere. So the proteases cannot come closer to that because there are sugars which they don't recognize. So if there are glycosylation, then it is very difficult to crystallize because they are moving all the time in the solution. They move. When they move, you can't crystallize. It has to be like solid like that. Then only all these molecules, similar molecules, get together and form a crystal. So it is a trial and error in a way. Yeah. question is like how we get the structure with the inhibitor. There are ways of doing it. One is called co-crystallization. You add the inhibitor to the protein. Um, Usually, people do like 10 times more. Like say, if you have 1 milligram of protein, no, it should be 10 mg per mil, 10 milligrams of milliliter solution. Then you add, say, 100 or maybe 20 or whatever, the higher concentration of ligand. But I have seen sometimes it is difficult. Then what I have done, I use 1-to-1. But very recently, we were just, because we couldn't get one drug candidate crystallized. So I use 1 to 0.2 in inhibitor. Because if the ones which is bound crystallize, it's enough for us. So it's again a trial and error, but you can use co-crystallization, or you can have a crystal, then you can soak. You can add same condition, which is crystal is sitting plus inhibitor in one condition, and then you add the take 1 drop of that into a cover slip and transfer the crystals into that and leave it for say longer period. Maybe sometimes 30 minutes is enough. In some cases 1 day is enough. Sometimes you need a couple of days. If sometimes it destroys the crystal, so then you can't do that. So you have to again find out which condition is suitable to do that. It's called soaking. The other one is called co-crystallization. Yes. Okay, so the question is, what is the suitable one or the easiest one or cost effectivity? And okay, I explained 4 methods. One is computation, which is nothing to do with the laboratory. Yeah, you need a good computer with good graphic card. Now it's in even this computer, it's enough. The other ones are the mostly abundant method, or many people are using X-ray crystallography because that's the well-developed method now we have. But there are problems with that. You have to have a crystal. If you can't get crystals, then you can use NMR spectroscopy. So there are experts for that. I can't do that because I don't know. NMR spectroscopy has a limit. That means you can go up to, I can't exactly remember, sorry, but a certain number of amino acids, like 150 amino acids or so, or less. Oh no, it's also 85 amino acids, I think, something like that. Not a big molecule. But in crystallography, if you can get a crystal, it doesn't matter. It's 700 amino acids, no problem. Or like 200 kilodalton, no problem. Right? But it has to be a one molecule, usually. Or maybe it can be several molecules. We can do that, like ribosome structure. It's many molecules. So it has been done with crystallography. But in nature, these big systems like Rubisco or whatever, these quaternary structures, it's very difficult to do with crystallography or NMR. Because of that, people are using cryo-electron microscopy more and more. So, but the problem with cryo-electron microscopy is that you cannot get high-resolution structures yet. Maybe like 2.5 angstrom structures, but in crystallography, we get like 1 angstrom structures. So it's very powerful details. You can see like carbon, and then the electron density is like around the carbon, and even that kind of tiny teeny details we can see it in X-ray structures. The cost, cryo-electron microscopy, you have to have an electron microscope. It's a huge system with nitrogen pumping or helium pumping, or it's very expensive, of course, but synchrotron business is more expensive, but we are users, so we are not going to do anything. But like as a company, we pay for the one in France, we pay 120 euros per crystal. So sometimes accidentally we pick ice crystals and we send it, okay, 120 euros, just waste. It happens. It always happens. So it's that expensive to collect data as a private one. But academically, they give you can do it free. Like in the European Union, you can do it free. But I don't think it's the other ones, they need the maintenance cost, right? It's very expensive to maintain these things because you always maintain vacuums, and a lot of electricity bill is very expensive now. So it's very difficult, it's very expensive. But for Max 4 laboratory in Sweden, we pay 52,000 Swedish krona. That means about like 5,000 euros per 8 hours. So we find it is cheaper. So it's just that one. But for the shipping and bringing the samples back, and so everything is costly. So it's very difficult to speculate, but to tell you. But crystallography is for the moment, most developed technique. Yeah. Yeah, sure. You can build anything in the sky, right? You can just say. But if I am in an editorial board of one of the journals, so I just send it to the reviewers, and whatever they say, I accept it usually, because I know those people. But if for a modeling, I would like to see more than 40 to 45%, more than that identity of the amino acid sequence they are using for the template. There's a template, and you just put the other amino acid into that template. So the identity of that should be more than 40%. That's the cutoff for me, and I will never accept any paper lower than that, because I can model, of course, with the low resolution, low identity, but I don't trust other people, because people are sometimes they don't know. They just give it to a server and get a model, and they submit it. It doesn't work like that. It's you have to do it by. If you do it by hand, then yes, I can think about it. But no, it has to be, because you cannot trust. Even you model with 90% accuracy, you can't trust. It's model is a model. It's not a structure. So the conclusion you obtain from that, it has to be very limited. You can say, okay, it has similar properties to that protein. You can say like that. You cannot say it has this for sure. No, you can't. Any more questions?
So thank you very much, sir. And I know there are some students from the environmental sciences domain here. So I personally would now go home and see how enzyme engineering can help in bioremediation. So perhaps my students, they might have now developed an interest on enzyme engineering, and maybe they could relate it to, in addition to drug discovery and drug science, we do have many applications of these aspects. Thank you, sir, on your enlightening talk, very educational, and I'm sure that it sparked a kind of a nice interest among our audience. And we were truly privileged to have you here for this talk also. And as a small token of appreciation, I would now like to invite our Dean, Sir, Professor Kumara, to hand over a small memento to make it a memory that you visited Sabra Sri Lanka. Thank you so much, sir. So even though we are very distant geographically, I'm sure that whenever we might need your assistance in the future, you will be all willing to do that. Thank you very much. So with that, let me formally thank you everyone for joining this third pre-conference event of the International Conference of Sabaragamu University of Sri Lanka, and on behalf of the Faculty of Applied Sciences. Thank you very much.
Sunlight fell and reminded me life can be so gracious sometimes. And I felt like everything around me was connected somehow. At night I hear the rhythm of the ocean as it breaks on the shore. And I think about all the things that I am grateful for. And they say, hold on to the ones you love, keep them close to you. And they say, hold on to this time we have, and let the light shine through. Sometimes I